Today, we will be going into apoptosis and processes specifically involving the mitochondria. Apoptosis can be divided into intrinsic and extrinsic apoptosis. Intrinsic apoptosis relies on a pathway inside the cell, triggered by a variety of internal cues. While they can come in many forms, the main catalytic signals are an increase in stressors, damage to DNA like a break in the helix, hypoxia, which is decreased oxygen levels, and survival factor deprivation like hormones that cells need to function. Extrinsic apoptosis is catalyzed from signals outside the cell. It is due to death receptors detecting extracellular signals called ligands that bind and induce cell death via a cascade of reactions. Ones of specific importance are the TNFR, tumor necrosis factor receptors. Both of these pathways can lead to apoptosis, but how do they do it? That's what we'll be covering today via the induction of MOMP, which stands for mitochondrial outer membrane permeabilization, via the activated proteins BAC and BAX. We'll be looking into the pathways as a whole, as well as how all the different steps in such critical processes are regulated on a cellular level. Terms that might seem specific to this pathway will be defined as we go, such as APAF1, caspase, cytochrome C, and the apoptosome. APAF1 stands for apoptotic protease activating factor 1. APAF1 is an adapter protein made up of a CARD, NOT, and WD domain, and it's responsible for oligomerization and construction of the apoptosome. But just how does it do this, you may ask? The APAF1 CARD domain, responsible for oligomerization and binding to caspase 9, and NOT domain, home to a nucleotide binding site of deoxy-ATP, are inaccessible due to the flanking WD domains. They can be made accessible by removing the WD domain, but not in this case. Instead, APAF1 changes conformation via the binding of cytochrome C, exposing the CARD and NOT domains in the active complex. We know that when lots of active APAF1 molecules collect, they form the structure of the apoptosome. However, the structure is not quite ready yet. So what's the missing piece, you may ask? That is caspase 9. And that, in this complex right here, is what we see in purple, just like we see cytochrome C in orange. Caspases are protein-cutting enzymes, also known as cysteine proteases, that cleave after the aspartate residue. They can be divided into executioner and initiator caspases. Executioner caspases are present in all healthy cells but in inactive form. They become activated when they are cut by initiator caspases, as seen being drawn now with 3 domains. Initiator caspases are activated when 2 identical chains are brought together by adapter proteins seen in red. Active executioner caspases go off and cleave substrates to initiate cell death. In our case, the initiator caspase is caspase 9, and it's activated by docking at the apoptosome via CARD-CARD interactions. Cytochrome C leads to APAF1 activation, leading to the apoptosome structure and caspase recruitment to achieve activation and full apoptosome formation. The apoptosome then activates caspase 3 and caspase 7, which are executioner caspases, which go on to cleave structural and repair proteins essential for cellular life. This is the general pathway of apoptosis induced by signal reception or internal cues. And you might recognize at this point that all of these downstream apoptotic processes are dependent on the presence of cytochrome C. But where does it come from? Without it, the CARD domain cannot be exposed, and the apoptosome would not be able to form, removing the activation of caspases 3 and 7 and avoiding apoptosis as a whole. With that, we look at MOMP, specifically via BAC and BAX, 2 proteins critical in mitochondrial outer membrane pore formation. They are part of a larger family of BCL2 proteins involved in apoptotic regulation. BCL2 can be divided into BH3 only, anti-apoptotic, and pro-apoptotic, with BAC and BAX being pro-apoptotic. BAX is free in the cytosol before its implantation in the mitochondrial outer membrane, while BAC is already latched to it. Upon activation, they enter the mitochondrial outer membrane and oligomerize to create a pore for mitochondrial components to diffuse out of. We know cytochrome C is there as it can be seen in green, and that is where it comes from to bind to APAF1. But what's in red? That's SMAC. SMAC is a key regulator of apoptosis and its ability to bind to ZAP, a caspase 3 and caspase 7 inhibitor, and rendering it ineffective. Essentially, what we are doing here with SMAC is inhibiting the inhibitor, as seen below, leading to functional caspases 3 and 7 and successful programmed cell death. Consisting of cell shrinkage and chromatin condensation, membrane blebbing, nucleus and organelle collapse, and formation of apoptotic bodies.